Welcome to today's lecture. As you know, we are in the series of fish related lectures and we are talking on the spoilage of fish. This is the second lecture. In the previous lecture, I have discussed the spoilage of fish due to enzymatic and chemical spoilage. Today specifically we will discuss about the microbial spoilage. So first we will see what kind of microbes present in fish and what are the reason for microbial contamination, and then we will understand mainly the bacteria involved in the spoilage, what kind of changes happen due to the bacteria, and some of the chemical details, the compounds produced, how it is produced you will see, and the impact of spoilage. So these are the plan for today's lecture. As I have already discussed in the previous lecture, there are three kind of spoilage of fish, in which the enzymatic mostly coming from inside, then oxidative, that is to some extent due to enzymes or due to the auto-oxidation. These two we have discussed in the previous lecture. So today we will discuss mainly the spoilage due to bacteria and other organisms, so we can call it microbial spoilage. Here first we will understand about the microbes present in fish or normal microbial load in the fresh fish just after catching. So in case of live fish or inside the mussel, normally it will be considered as sterile, so no microbes will be there inside the mussels, etc. However, the microorganisms can be found on the outer surface and also inside the alimentary tract, even in live fish, and this number can be in a variable number, particularly in newly caught fish. So the normally number of microbes which we can find is around 10 power 2 to 10 power 7 CFU per centimeter square on the surface or on the skin. It can be up to 10 power 3 to 10 power 5 in the gills, and about 10 power 9 to 10 power 10 CFU per gram in intestine. So this is a normal microbial load, but when it is dead for long time, these numbers will heavily increase. So this is in case of fresh condition just after catching. So now we will see mainly the bacteria present in the fish, but what is its action on the fresh fish? As we have told in the previous one, there are millions of bacteria present on the outer surface or inside the fish, particularly in the intestine. But when it is alive and healthy, so there is a natural protection against harmful bacteria. So there is a natural defense mechanism. Because of that, no bacteria can act on the fish or no adverse action will take place. That can happen only when there is a failure of defense and immunity, and that lead to a starting of disease. So normally healthy fresh fish, the bacteria will remain only on the surface or inside the GI tract. It will not have any effect. So in live fish, bacteria present on the skin, gills, intestine, but cannot attack the fish muscle. Before we discuss about the spoilage of fish, let us understand the contamination of fish. Contamination means the microorganisms coming to the fish, either from the intestine or from outside different sources. So when the bacteria is reaching the fish or the flesh of fish from other sources, that can be considered as a contamination. Contamination means mostly about different kind of microorganisms like bacteria, fungi, yeast, mold, virus, and many other toxins or other toxic substances. So the sources of contamination, mainly it is from the water. So if the water is contaminated, that will provide the more organisms or other toxins, etc. on the fish. Then from intestine, so when it is dead and unprotected, the intestinal bacteria will enter into the muscle by invasion, as I mentioned earlier. Then at the time of catching, so at what time fish caught or how much there is a delay, that makes a factor for contamination, or due to other appliances where it is kept. density of microbes in the surface. So if the surface has got very high density, naturally that's a major source of contamination for the mussel. Then from the equipment or appliances, as I mentioned, from the storage, or if there is improper storage or unclean vessels or containers, that can be a source. And during transport, different vehicles used, that also can be a source of contamination. Now briefly we will discuss about the sources of contamination. The first is the water. So in case of freshwater fish, it will carry the freshwater bacteria, because the nature of bacteria will be different in freshwater and the saline water or sea water. So in the northern water carry mostly psychrophilic bacteria. That means that low temperature conditions, the psychrophilic bacteria will be present in the water, whereas in the tropical region the mesophils bacteria will be present, that is around the temperature of 20 to 35 or 40 degrees Celsius. Then at the time of catching, that is a sources of contamination. Firstly, the number of microbes on the surface influenced by method of catching. So method of catching is a factor for contamination. Similarly, the injury during catching, if there is any injury, that will lead to quick contamination. Then another source is intestine, as I told. So both saltwater and freshwater fish will have huge microbes in millions in the intestine, and if there is a delay after catching or after dying, then those bacteria will go to the mussel by invasion from the intestine. In continuation about the sources of contamination to fish, which leads to the spoilage. So density of microbes in surface slime. So if, as we all know, there will be a slimy layer on the surface, and if the microbial load is very high, naturally it will be a good source of contamination for the fish inside or the mussel of fish. From equipment, so the kind of equipments involved in the catching, like boats, boxes, fish house or fisherman himself and other appliances, they are the good source of contamination. So if they are not hygienic, then from fish storage, so contamination can happen from the low quality of ice or ice box or other storage devices. If those devices are not very hygienic or sanitary practices are not followed, then they are the source of contamination. And finally, the transport. At the time of transporting, contamination may start, particularly if it is without ice or without cold temperature. Then all the appliances touching will give a contamination, particularly if they are not very hygienic. Similarly, during transportation, contaminated fish may contaminate the fresh fish. So already some fish is there old and dead, and then we keep the fresh fish, so there will be a cross contamination. Now we will discuss about the microbial spoilage. When we say microbial spoilage, it includes bacteria, virus, yeast, mold, everything. Now first of all, the composition of microflora on the newly caught fish depends on the microbial contents of the water in which it lives. As I discussed previously, the sources of contamination. So in case of fresh fish, majorly it is the water. What kind of microbes present, that only comes to the fish. Then secondly, it comes the other factors like handling and other appliances, as I mentioned. So depending on all those, the profile of microbes will change, whether it will be what kind of bacteria or other organisms. Now microbial growth and metabolism is a major cause of fish spoilage. So once the microbes are there, once the fish is dead, all kind of microbes will grow, and that will produce different kind of chemicals, and that leads to off odor and spoilage. So firstly, there can be biogenic amines such as putrescine, histamine and cadaverine. Then there can be organic acids, sulfides, alcohols, aldehydes and ketones, and all together they produce unpleasant and unacceptable off odors. So then we call it spoilage. So all these details step by step we are going to see later. Now we will see the bacteria causing spoilage. So in the microbial spoilage, most important is the bacteria. Majorly they are responsible for spoilage, so we will call it bacterial spoilage. There are three categories mostly depending on the temperature. When the temperature is low, that is at chilling temperature, the common spoilage organisms are Pseudomonas, Achromobacter and Flavobacterium. Then at normal room temperature or ordinary atmospheric temperature around 20 to 35 or 40, at this temperature we call it mesophils. So mostly Escherichia, Proteus, Serratia, Sarcina and Clostridium. So these kind of organisms are the responsible for spoilage of fish, and at a higher temperature those are called as thermophils or thermodiuric organisms. So they are like Micrococcus and Bacillus. They cause the spoilage to fish. Here we will see quickly the mode of bacterial action. So bacteria which is there on the surface or intestine, how they are reaching to the muscle and acting. So once the fish is dead, the bacteria from the skin slowly will penetrate to the muscle through the skin, because the skin will have the permeability once it is dead. So this will be more fast if the skin of the fish is thin. Similarly, once the fish is dead, the intestinal bacteria also will enter the flesh, because once the fish is dead, the natural barrier which prevents the entry of the intestinal bacteria to the muscle is lost. So the natural defense is lost. So the bacteria will enter through the membrane and will reach the muscle. Now the bacteria coming either from the surface or from the intestine will start multiply very quickly, because these muscles are excellent source with reach of nutrients, and it gives very quick multiplication. And at this process the bacteria will produce different kind of metabolites, as I have mentioned some of the name of the chemicals earlier. So all these are the reason for the spoilage. So the bacteria and enzyme, because of their activity, produces different kind of undesirable compounds, and which leads to the change in fish odor, flavor, and that flavor can be sour, gassy, fruity, and finally ammonia or fecal odor. So all these are undesirable and unacceptable, and then we call it spoiled. Now we will see the effect of bacterial growth. Due to the bacterial growth and production of metabolites and the action of different kind of enzymes, so there will be a slime layer formation due to the metabolites. There will be discoloration of gills and eyes, and there will be a loss of muscle texture. This happens in an advanced stage when there is breakdown of protein. So due to the bacterial enzyme, there will be proteolysis. So the muscle will lose the texture and it becomes soft. That we can check by pressing with finger. The volatile compounds from the putrefaction of protein. So when the proteins undergo very advanced changes or deterioration due to proteolytic bacteria or proteolytic enzymes, so then it will produce different kind of off odors, such as fishy odor, which is due to the formation of trimethylamine, and also it can produce putrid odor due to the advanced spoilage of protein. So bacteria such as Salmonella putrefaciens, Acromonas, Enterobacteriaceae, Photobacterium and Vibrio. These are some of the common bacteria which produces ammonia-like off odor or fishy odor. So further details we are going to see next. In continuation about the effects of bacterial growth, as I mentioned, the Pseudomonas putrefaciens. It is a fluorescent Pseudomonas, and other spoilage bacteria increase rapidly due to their fast multiplication, and they produce many proteolytic and hydrolytic enzymes or lipolytic enzymes. So naturally they produce different color changes, like the greenish yellow color on fish caused by Pseudomonas fluorescens, yellow color changes by Micrococcus, and red color changes by Bacillus and Sarcina. Similarly, there can be a chocolate brown color caused by yeasts and molds, and a musty odor by streptomyces. So these are changes by the yeast and molds. Here we will see some of the common spoilage microorganisms and their corresponding what kind of chemical compounds they produce. So first one is Salmonella putrefaciens. It produces trimethylamine, hydrogen sulfide, methyl mercaptan, dimethyl sulfide, hypoxanthine and ammonia. This another common spoilage organism is Photobacterium phosphorium. This produces trimethylamine and hypoxanthine. Then there is Pseudomonas species which produces ketones, aldehydes, esters and hydrogen sulfide. There is Vibrionaceae species. They produce trimethylamine and hydrogen sulfide. There is aerobic spoilage organisms. Generally they produce ammonia, acetic acid, butyric acid and propionic acid. Then there are lactic acid bacteria. Commonly there is a group of organism which produces hydrogen sulfide, ketones, esters, aldehydes, ammonia and acids. And finally the yeast and anaerobic rods. Anaerobic bacteria, they produce ketones, esters, aldehydes, ammonia and acids. So these are some of the common spoilage organisms and their metabolites. Here once again some of the common spoilage organisms and the metabolites they produce is highlighted. Most importantly Pseudomonas, which produces ketones, aldehydes, esters and sulfides. Then the Phosphobacterium phosphorium, which produces trimethylamine and hypoxanthine, which is very common for surface reaction and producing the fishy smell, this trimethylamine, which I am going to talk more details. Then anaerobic spoilage organism produces ammonia, acetic, butyric and propionic acid, and Vibrionacea produces trimethylamine and hydrogen sulfide. Now we will try to understand the deterioration process in fish, particularly the chemically how the changes happens. So in the fish muscle we can expect mostly proteins and lipids. There will be very little carbohydrates. And all the deterioration happens by three ways, as I mentioned in the first lecture and today at the beginning. One is purely chemical, another is due to enzymatic. From the normal enzymes present inside the muscle, after death they become active. And today majorly we are discussing about the bacterial action. So due to the chemical action, as I discussed in the previous lecture, that will lead to autooxidation and breakdown of fatty acids leading to rancidity. Whereas hydrolysis also can happen due to hydrolytic enzymes either present in the muscle endogenous or due to the bacterial enzymes which can be lipolytic. So they cause breakdown of lipid and hydrolysis leads to a kind of rancidity, as I discussed earlier also. And the major changes happens here in the protein. Again this can happen due to endogenous enzyme or due to the bacterial growth and bacterial enzyme. So when the protein is broken, it will be forming the peptides and amino acids. Further there can be deamination of amino acids and it will form ammonia, and there can be decarboxylation of amino acids and then it will form carbon dioxide, and both the process will produce different odoriferous compounds which I have told at the beginning. Here once again we will try to understand the chemistry of bacterial spoilage. So due to the bacterial growth, a lot of enzymes will come from the microorganisms and that act either on the protein, carbohydrate or fat. In case of protein, the proteolytic enzymes from the microorganism, it will break it and form amino acids, amines, ammonia and hydrogen sulfide. Now this ammonia and hydrogen sulfide will produce the undesirable odor effect. Similarly from carbohydrate, though it will be very less, that will undergo the fermentation due to the specific organisms, and that will produce acids, alcohols and gases, and that produce the sliminess, and the fats will undergo the fatty hydrolysis or lipolytic changes due to the lipolytic microorganism or lipolytic enzymes, and then it will produce fatty acids and glycerol. Here we will understand a very important chemical change in bacterial spoilage of fish, which is not seen in case of animal origin meat. It is only specific in fish muscle. So this is the reduction of trimethylamine oxide to trimethylamine. So normally fish will have a outer layer which will have trimethylamine oxide that undergo a chemical change by reduction and forms trimethylamine. So normally this trimethylamine oxide will not have smell, whereas trimethylamine will have the smell. That is one of the main cause of fishy odor. So this TMA level is commonly used to determine the microbial deterioration leading to spoilage. So fish normally use this TMAO as an osmoregulant to avoid the dehydration in saline water. So because the outside water is highly saline, so it will always face a dehydration. So water from the inside will come out. So to prevent that, it is having a coating of TMAO. Similarly, in case of freshwater fish, it works in a other way. When the fish inside has got more waterlogging, that will have a reverse effect. To protect that, they will have a TMO, but this is more concern related to marine fish. Now the some bacteria like Salmonella putrefaciens, Aeromonas, Enterobacteria, Phosphobacterium, Phosphorium and Vibrio species. All these bacteria will act on this TMAO and convert to TMA, and in this process it can get some energy and produce ammonia-like of odors, and that is the fishy odor due to formation of TMA. So in continuation to chemistry of spoilage or bacterial spoilage in fish, so as I mentioned, the protein will break down and forms peptides and amino acids due to the bacterial enzymes. Now further, these amino acids can undergo breakdown and formation of primary amines. For example, there can be formation of histamine from histidine, formation of arginine from glutamic acid, etc. So these bacterial action may cause sometime food poisoning in extreme cases. Similarly, there can be breakdown in urea. The high concentration of urea in flesh of some fishes is degraded to ammonia by microorganisms. So some fish will have higher level of urea which will be broken down by the microorganism and produce ammonia, and that formation of ammonia can have offensive odor during spoilage. So we have discussed the microbial growth and chemical changes leading to spoilage due to the undesirable products or metabolites. So we will see the finally now when the fish is spoiled, what is the impact. So what are the signs? So first of all, there will be discoloration due to spoilage. There could be yellowish to greenish discoloration caused by Pseudomonas fluorescens, Micrococcus and others. There can be red or pink discoloration due to Sarcina, Micrococcus or Bacillus species, or sometime by yeast and molds. And there can be chocolate brown discoloration due to asparaginous yeast. So precisely these are some of the common discoloration happens due to microbial spoilage of fish. Now here we will see the final impact of fish spoilage. So there can be putrefaction due to the extensive breakdown of protein, and there can be slime. There can be sourness due to production of acids due to the action of lactic acid group of bacteria and also leading to discoloration. There can be rancidity either due to autooxidation, oxidative spoilage, or there can be due to rancid odor by lipolytic changes or hydrolytic oxidation or rancidity. And sometimes there can be other infection. So ingested with when the contaminated food with bacteria multiply and produce toxin in the consumers, then cause illness. So that's the infection, and sometimes there can be intoxication. So many organisms which grow in the fish and produce toxin, so they may die, but toxin will be there when we consume the fish, and that also can cause illness. So that is called intoxication. So now we are at the end of today's lecture. Today we have discussed about the microbial spoilage of fish. So first we have discussed the normal microbial profile, and then how the contamination happens, what are the factors. Then we discussed briefly about the mode of microbial action and how it causes spoilage due to the production of different metabolites. We have discussed about the common microorganisms and what kind of chemicals it produces, and then we have seen the different kind of process happens due to the microbial growth and some of the chemistry aspect, how the protein, lipid, fat undergo the changes and leading to undesirable deterioration and spoilage of fish. So in short, the most of the spoilage in fish is due to bacterial breakdown, though there is the other aspect like oxidative and enzymatic spoilage, which I discussed in the previous lecture. One important characteristic in fish spoilage is that the formation of trimethylamine, which is not there in other mussel foods. So this gives the typical fishy odor, and this odoriferous amine is responsible for the smell. And moreover, in case of fish, there will be lipids, particularly fatty fish. So those fish will have hydrolysis due to the bacterial enzymes, and earlier I told due to the endogenous enzymes also. And particularly many fish will have polyunsaturated fatty acid that undergo quick oxidation. So to avoid all this, there should be chilling of the fish immediately after harvest as early as possible. That's the preservation which we will discuss separately in different lectures. So thank you for your attention.